Good morning. Thank you, Zia, for that introduction. I'm delighted to be able to talk with you today about lung regeneration and discuss together for the next 40 minutes whether this might be an achievable mission. I'll touch on two themes today. One is to tell you the story of our now 13-year quest to try to derive de novo lung epithelial lineages, tissues, or bioartificial functional entire lungs from stem cells. And I'd like to speak about the future and whether lung regeneration and what President Obama calls precision medicine might be part of that future using stem cells. This is an ATS discovery series, and so I'd like to highlight two discoveries during the next 40 minutes that I believe are key and transformative for biomedical research and the hope of our patients. One is reprogramming to make iPS cells, and the second is the ability to now edit the human genome. So I think achieving regeneration of the lung is a watershed moment for us. We are currently in an era which I believe is defined as helping the lungs repair themselves, doing no harm. We have made miraculous advances in mechanical ventilation where patients are sustained, living longer and given the chance to heal their own lungs. And the era I'd like to move towards and visualize that future with you in the next 40 minutes, I'd like to move towards an era of augmenting repair, increasing the ability of the lungs to heal themselves, and perhaps even engineering regeneration. And by that I mean helping the lungs reconstitute, heal after injury, and perhaps even delivering new reconstituting cells or tissues or even autologous entire lungs to our patients. So with that idea in mind, when I opened my lab in 2002, 13 years ago, we began to visualize this mission. This is captured by Finn Hawkins, who's drawing here the idea of taking a stem cell and coaxing it in a laboratory dish into lung epithelium via an identifiable lung progenitor, which we could only imagine at that time point. And we chose to do this work with pluripotent stem cells using the, at that time, gold standard of pluripotent stem cells, the embryonic stem cell population. This is in the days before iPS cells were discovered. And why we chose to use embryonic stem cells to engineer lung, we thought might be impactful if we could generate cells that might teach us something about the way human cells behave, get lung disease, or respond to therapeutics in a personal way. And one day we might even engineer reparative cells or tissues or entire lungs. And so I need to first go back to the beginning to explain to you what is pluripotency and what are these gold standard embryonic stem cells and why are they important. And by the beginning, I mean your beginning here on the left, where you are a fertilized egg at the moment of conception. And do you remember that glorious day when you divided once, twice, thrice, and you arrived here at this ball of cells called the blastocyst embryo, pre-implantation in the uterus. And in this ball of corner here called the inner cell mass, all the cells that gave rise to you at this very moment, all of your, including all of your lung cells, originated from all these very few precursors that are without doubt scientifically, unquestionably pluripotent. But these cells are not stem cells. They are fleeting, they make cell-fate decisions, and they differentiate within hours of this photo. And there's no evidence that we have leftover pluripotent cells of this phenotype once we are born. And so the amazing discovery in 1981, Gail Martin and Evans published the technique to capture these cells in stable state in culture, and that is the invention of embryonic stem cells. Shown here, colonies of 20 or 40 or so cells growing in culture that were grown out of this inner cell mass, and they are now captured in a stable state. And so what I've just said to you is that embryonic stem cells represent the discovery of how to capture in culture a pluripotent state that is normally transient in nature, and these cells are now stable and self-renewing and may be called stem cells. And so now that I've told you about those cells, we can go back to our mission and try to envision how those cells that we know are reminiscent of the blastocyst embryo's developmental stage, how we turn those cells into lung cells might be most efficiently performed by copying the way nature does it, the way we developed in our 9 months of gestation as we developed lungs from similar precursors, and trying to recapitulate that process in the laboratory dish might be a worthy goal if we are to succeed in this mission. So then the first question to ask is, where does your lung epithelium come from? In studying developmental biology, is part and parcel of achieving this work. So your lung epithelium comes, if you look in a textbook, you'll see pictures like this. Your lung epithelium apparently comes through a branching, budding iterative process that also involves sequential differentiation steps. And if you rewind the clock backwards, you could see that your lung budded off of the foregut endodermal tube. But I would say this is not early enough. And like physicists or astronomers, in my group, we think of those people that rewind the universe all the way back to the Big Bang when it all started. And we need to go earlier than this moment. And for us, the Big Bang of lung developmental biology is represented in this schematic. So here you see, you can imagine the foregut endodermal tube. And just moments later, these cells that are endodermal undergo a cell fate decision that's really remarkable and miraculous and mysterious still. It's a decision signified by induction of this locus, NKX2.1, or for this audience, perhaps better known as TTF1, thyroid transcription factor 1, the canonical lung cancer marker. It's a locus encoding this key transcription factor required for lung development. And induction of this locus marks the moment, the actual conception of a cell fate decision for a gut tube cell to commit to a lung epithelial cell fate. It's also expressed in the endodermal gut tube in the thyroid domain. Those are the only two places in endoderm this gene is expressed. And so there's nothing quite like seeing the actual data from developmental biology represented here with some in situ hybridizations, these two performed by, in Wellington Cardozo's lab by Tushar Desai. And you can see a foregut tube is hidden here. It's just a foregut. Up here, the thyroid is specified, and just hours later, 9 days after fertilization, this bright purple signal shows the specification event by in situ hybridization of NKX2.1 induction, and these cells have committed to lung cell fate. And we'd like to understand this window better as a way to start to engineer cells. And so to do that, we understand that we first need to make out of stem cells the best possible foregut endodermal cells we can make. So we studied the work of Gordon Keller, who pioneered a phrase, directed differentiation, which is the recapitulation of known in vivo developmental milestones to guide pluripotent stem cell differentiation. And part of achieving this mission, then, we knew, was to try to make definitive endoderm or foregut endoderm on our way to achieving lung. And studying Gordon Keller's work, we found that induction of nodal signaling with the growth factor activin can make stem cells differentiate in culture. So this is a flow cytometry picture day by day showing you the beautiful kinetic of how cells differentiate that start from mouse embryonic stem cells in the pluripotent undifferentiated state and end up as definitive endoderm. And by now you could see from the calendar today about 8 years have passed as we practiced making the best definitive endoderm we possibly could. And the details captured in this paper in the JCI by my graduate student Constantina Christodoulou provide the details of how, and microarray databases that say that these cells are very much like embryonic definitive endoderm that we all naturally made during gastrulation. So we are ready now to go on to the task of actually inducing that big bang of lung development, the lung cell fate decision in culture from these stem cell-derived endodermal cells. And we were actually not very good at doing that. We had no success until we met this scientist named Hans Snook, who serendipitously discovered a technique. He was trying to derive thymus, thymic epithelium that comes from the foregut, and he discovered two inhibitors of BMP signaling and TGF-beta signaling applied to these stem cell endodermal cells, patterned them to foregut endoderm. And so then working with Hans really helped us go on to the next step to try to induce lung. So we're now ready, having patterned the stem cells into foregut endoderm, to try to induce the cell fate decision to become lung, this very difficult decision. But what we realized was we knew nothing about these cells. You can't find literature really on their phenotype at the moment they're specified, and we need to understand those cells and their programs in order to recapitulate them. We also would like to know in our patients' lungs, are there any leftovers of these cells, or are they recreated during injury repair, or could they be harnessed for regeneration? And so to learn more about these cells, we built this tool. It's a GFP reporter, green fluorescent protein knocked in to the NKX2.1 locus in the mouse genome. And we do that, knock in embryonic stem cells, and then we make mice out of those stem cells by blastocyst transfer. And here's a picture of the mouse. You could see the beautiful bright green trachea, the lung buds just starting to bud at E9.5, 9.5 days after fertilization. Here's the thyroid that's green, perhaps not projecting well for you. And here's the forebrain, the 3 places in the embryo where this transcription factor is expressed. And so the task then was to try to take out these lungs and wind back the clock and see how the lungs developed from the very earliest inception of lung cell fate. And the pictures of those lungs are shown here. You can see as we go back in lung development, you can actually see the lobes starting to bud off of the respiratory tree, 11.5 days of development in this mouse. And if we go back even further, past the primary lung buds, to that very moment when a foregut endodermal cell committed to lung epithelial cell fate, you could see that captured with the green fluorescence here. And just 12 hours earlier in that foregut tube, you can't see anything. The foregut has changed from here to here. And so what we have in essence established is the moment of cell fate decision that you could witness with the induction of green fluorescence in this model system. And now we might learn something about these cells. There are just 100 of them. They're very rare. And Lairs Economou spent 5 years dissecting out of mouse embryos, hundreds of embryos from this green domain compared to the pre-specified endoderm. He sorted those cells, analyzed them with RNA-seq, and in collaboration with Avi Spira's group at Boston University, Laertes has produced what he likes to call the genetic program of the first primordial lung progenitors. And this is an essential database for us. It is a roadmap that guides us on how to do this work and how we might now engineer the same cells in the laboratory dish. So after several years of inducing lung, we successfully, guided by the roadmap of in vivo lung development, we successfully formed in a laboratory dish a system where embryonic stem cell-derived endodermal cells could be exposed to defined factors that induce signaling pathways that let cells behave like this, I think summarized in this movie. If it projects, you could see endodermal cells that here a green event will come up in front of your eyes. There it comes now. And I'm not sure if you could see, but a field of green cells starts to emerge. And this is mouse lung or thyroid NKX endodermal specification occurring in front of your eyes. And this short movie would summarize, I think, 7 years of work in my laboratory and is one of our proudest accomplishments to date, and I really enjoy showing it to you. It's totally worth it. So the publication of this work in 2012 now established a sequential recipe, like cooking, where in the laboratory dish you could make endoderm out of mouse embryonic stem cells and then induce the lung cell fate decision, and you could use machines like flow cytometers to purify those cells and culture them further in pure form. And we had some feeling of their differentiation repertoire as the primordial progenitors then upregulated some transcripts that we recognized as indicating lung epithelial programs, such as surfactant protein C, SPC, or the club cell marker CC10. Now, we also began to understand and define the global transcriptomic programs of those in vitro engineered cells, and we started to understand the cell fate decision that those endodermal cells made as they committed either to being this NKX endodermal cell or a non-lung, non-thyroid cell. And we found that there was cellular memory in the system, that the cells were able to pass on to their descendants memory of this cell fate decision so that they wouldn't misbehave. Just like your elbow stays your elbow for your whole life and your lungs stay your lungs, and there's really no elbow to be found in your lungs. And this is a cellular memory process that is complicated but is encoded by an epigenetic landscape that changes, and we've made some progress in mapping and trying to understand the epigenetic landscapes and defined histone modifications that encode that cellular memory and are passed on to the descendants of the cells that have made the cell fate decision in vitro. Now, this is perhaps some progress, but I know you are thinking these cells don't look like what you would call a lung. They're green and they're flat in a monolayer, and the lung is really defined by its function and structure, its three-dimensional special structure. And so we are ready to take our cells and start playing with a way to make three-dimensional structures that might be more reminiscent of what a pulmonologist would call lung. And at that time, we met Harold Ott, who had invented a technique called perfusion decellularization to create nature's scaffolds of organs that might be used for tissue engineering. And so I'd like to explain to you Harold's invention and how we then used his invention to try to generate engineered lung tissue from stem cells. So Harold's invention is a seminal and pioneering publication represented here. He's a futuristic, advanced thinker who isn't scared of 50 or 100-year milestones to engineer lung tissue. And it started with this important paper where he has perfused rat lungs that then go clear white as just the scaffold remains and the cells are stripped away. Now, in this paper, Harold took rat-digested cells, including epithelial cells, and perfused them down the trachea of this scaffold to re-epithelialize the lining of the lung, airways, and alveoli. And down this pulmonary artery catheter, he infused human umbilical vein endothelial cells. And in a bioreactor, when this graft matures, he was able to ventilate the structure and perfuse blood through it to measure gas exchange. And remarkably, the bioartificial lung, as he and I like to call it, could be transplanted into pneumonectomized rat recipients. So this is a movie Harold took of after pneumonectomy. This rat has this bioartificial lung generated here in the bioreactor with ligation of the bronchus, pulmonary artery, pulmonary vein, and the lung is functioning in the rat's chest compared to the native right lung here. And Harold was able to perform this fluoroscopy to show function of the graft. So you can see aeration of this bioartificial lung with the diaphragm going up and down. And we were fortunate to participate in collaboration with Harold on this first paper he published in 2010 for this technique. So with this technique, we are now ready to test, instead of rat-digested lung cells, the cells we had engineered that I showed you in the movie, the mouse embryonic stem cell-derived candidate lung progenitors, isolated by flow cytometry in pure form, now irrigated down this trachea of a decellularized mouse lung. And the histology was interesting. It was starting to look like three-dimensional alveolar structures. Then some of the cells in those structures were expressing the NKX 2.1 nuclear protein, and other cells were losing that protein while they flattened out, reminiscent perhaps of the shape of type 1 cells. And this is a pure epithelial type prep infused into this graft. There are no endothelium or interstitial cells here present, so I think it gives you a feeling of how far we've come, but also how far we have yet to go, where we're missing many lung lineages from this model, and it will take a long time to, I think, advance this work. Now, I've been speaking to you about mouse cells up to this point. Many of us are doctors or human researchers, and so I think the teaching point here is that the mouse model for us was instrumental. We discovered the pathways and the recipes in the mouse system, and the literature we read was not human. It was actually frog, xenopus, worm, and Drosophila literature to come up with these recipes. But we're ready now to apply that biology, because all biology is consistent and all research is translational, I believe. We're now ready to apply this biology to derive human candidate lung epithelial cells. But we realized there were two key issues that would limit this kind of progress. One is that even if we could make lung progenitors, or whole lungs, from human embryonic stem cells, those lungs would be allogeneic still to our patients and would be rejected after transplantation. And we knew also that the GFP reporters that we had used to advance our mouse work couldn't be engineered easily into human genomes. There's difficulty in manipulating the human genome up to that point. So these two seminal advances I'll introduce you to really helped us surmount those hurdles. The invention of reprogramming to make autologous cells and tissues from our own patients, and the invention of genome editing that allowed us to generate some reporters I'll show you. And so the first discovery of reprogramming is summarized in this paper. This is Shinya Yamanaka's Nobel Prize winning Cell paper from 2006, where he published 4 transcription factors, OCT4, SOX2, KLF4, and c-MYC, transiently introduced into these cells, somatic cells that were dermal fibroblasts, could reprogram these cells and reset their entire chromatin and transcriptomic programs all the way back into a state that looked very much like an embryonic stem cell. But because they weren't derived from embryos, Dr. Yamanaka named those cells induced pluripotent stem cells, which share a similar transcriptome, epigenome, and functional repertoire to embryonic stem cells. They're highly, highly similar. So to understand what iPS cells are and how good and important they are, I'd like to show you a movie I took in my lab where we've generated with the Yamanaka factors iPS cells by reprogramming the tail-tip fibroblasts from a black mouse. And those fibroblasts are now endowed with pluripotent properties they wouldn't normally have. And we can test for those properties in this very easy to understand functional assay where we transplant the black reprogrammed tail tip fibroblasts into this blastocyst embryo, which is taken from an albino host. So here come the cells into the blastocyst embryo, which is held in place by the suction catheter. And as the black candidate iPS cells come into the white or albino host embryo, we can now take this blastocyst and transfer it into a foster mouse mother, just as has been done for embryonic stem cells for many years, and we can watch what is the contribution of the mouse pups that are born that derive from these transplanted blastocysts. So let's take a look. 18 days later, those pups are born. A few days later, they get hair, and we can look at them, and they appear like this. These are mouse mosaics where you can see the black coat colors deriving from the injected reprogrammed cells on the background of the host albino mouse. And you can imagine that similarly all the internal tissues of these mice are chimeric, including the germline, the eggs and sperm. And you might test for that possibility, for example, by breeding one of these chimeras together with an albino host mate and simply looking at the next generation for coat colors that are black for evidence of germline transmission. So let's do that breeding. Go ahead and take a look at the next generation. And one can see here the easy to understand evidence of the pluripotency of the cells that derive from a tail tip fibroblast but are now pluripotent, equivalent to embryonic stem cells, including the capacity to transfer through the germline. And if you just imagine with me for a moment that these mice and all their lung cells, including all their epithelial and vascular and interstitial cells, are derived by reprogramming fibroblasts, you can really start to dream about what this might have to offer our patients if we could take this experiment and start to adapt it to human model systems. And I get quite emotional showing this picture because I remember a time that Gustavo Mostislavsky and I saw these mice and realized life would be too short to accomplish that goal, but we would certainly have a lot of fun trying. So let's see how that reprogramming methodology now might work in human cells. Remarkably, it works very similarly, and you can take the same Yamanaka factors, OCT4, KLF4, SOX2, and c-MYC, and by transient expression in human fibroblasts now, taken from a child who had a devastating lung disease from an NKX2.1 mutation, we could generate iPS cells. And these are very similar to ES cells. They will grow forever in the laboratory in the undifferentiated state, and they will live on long after we are all gone. They carry the mutation that the child had that caused the lung disease, and they are linked to the clinical phenotyping data of that patient. For example, their CT scan, lung biopsy, and many other clinical parameters. And so these cells might have something to offer biomedical research, and in fact, we've generated a large bank. We published more than 100 patient-derived lines generated from individuals with lung diseases, and some of the monogenic causes of those diseases I've shown you here, and the cells are in the bank and available for open source sharing with the world, and we've shared these cells already with many in the ATS community. And you might consider that these cells they don't have much to offer yet unless we're able to differentiate them into lung cells. So as the freezers fill up with these cells, I'd like to discuss with you how we might start to engineer human lung cells from these patient-derived stem cells. And I'd ask you just to consider also that these cells can now be made from a teaspoonful of peripheral blood. We no longer need to skin biopsy our patients to achieve iPS cells. So I think I'd remind you that in the mouse, we could induce the lung cell fate decision using these very valuable reporters like the GFP knock-in reporter that would indicate the lung cell fate commitment in response to these signaling pathways. And we're really at a loss to do the work in humans without a similar NKX2.1 GFP reporter that we might follow as being active in the human genome, like a traffic light telling us the moment of human cell fate specification. The good news is it's now able to engineer those reporters with the invention of human genome editing that I'd like to introduce you to now. I'd remind you that for years, scientists have been able to read the human genome, that is, sequence it. They've been able to write the human genome, that is, use PCR to generate nucleic acid sequences, but we've been unable to rewrite the genome or edit it. And scientists working around the world have discovered that certain nucleases, like this one shown here, when fused to certain amino acid moieties, like zinc fingers in this picture, those moieties and certain sequences can target the nuclease to a specific place in the human genome. And so genome editing works in a nutshell, by introducing a double strand break, or nick, into a very defined spot in the genome that you could pick at will. And that nick mobilizes a machinery of repair to that spot in the genome, and in the presence of a donor template that you could engineer and put in the cell, such as a GFP reporter, the cell can introduce for you that template into the genome precisely at the location you wanted to edit. So this is the first generation of gene editing, zinc finger nucleases, and there are now 3 generations that are popular, including with names like Talens and CRISPR-Cas9, but they all work the same way by nicking or cutting the genome at a specified locus to deliver a DNA template. So I'd like to introduce you in the next few slides to some experiments we've done with Brian Davis, where we've engineered that NKX2.1 locus that I showed you in mice, and we've engineered in humans this double-strand break using the Talens targeting system, and in the presence of the GFP reporter, through homology-directed repair, we've been able to introduce the GFP reporter right into the human NKX2.1 locus in iPS cells, and this schema shows how Philip Kramer in Brian Davis's lab cut in the second intron of this gene to put the GFP reporter into the NKX2.1 human locus. So the cells are now really powerful. They're really informative because we can start to do very fun experiments in the laboratory where we start to induce in human iPS-derived endoderm the green reporter. And Finn Hawkins, here to give you a feeling for how he does his work, will take many wells and play with recipes, timing, dosing of the same growth factors that work to induce mouse lung that I showed you. And from a flow cytometer, he can pick out certain conditions. And at the end, working in collaboration with Hans Snook, we have developed an efficient, reproducible human NKX2.1 induction protocol that mimics the mouse biology and produces cells that I'd like to show you in the next slide, which are generated now from a cystic fibrosis patient whose skin fibroblasts were reprogrammed into iPS cells that we then turned into endoderm and foregut endoderm. And then using very similar signaling pathways to that we used in mice, we generate green cells in a 3-dimensional culture system that look like this. So these cells are very pretty. They're monolayered epithelium. They're all green. We would like to find out whether they actually represent something akin to the first primordial lung progenitors specified in human development. And I have another movie to show you that will show how these cells grow in a dish. So in this movie, you can see cystic fibrosis iPS cells derived from a child's fibroblasts, reprogrammed, changed into endoderm. And in this 3D culture system, some of these monolayered endodermal epithelial cells are turning green. And for me, it doesn't get any better than this. This, to me, is watching the moment, the Big Bang moment of human lung lineage specification in front of our eyes. Next slide. So Finn Hawkins has used this system now, previous slide, yep, to purify just those green putative lung progenitors out, next slide, and defining the global transcriptomes of these green cells over time. Next slide. And just to select out the lung epithelial markers from this data set, you can see that over time, compared to real 21-week fetal lung epithelial cells, SPC-positive distal lung epithelial cells purified by Susan Gutentag from a human in these 3 columns, that our cells over time start to move towards that goal, as these red colors show the induction of these lung epithelial markers. And this panel shows both how far we've come and how far we've yet to go if you just consider that some markers, canonical markers like surfactant protein C, are quite cool here. Blue is far from the real lung. And we've developed some hypotheses for why that might be. You might consider that all the cells we're growing are expressing SPC at too low levels. They're not quite mature. But the hypothesis I prefer that we'd like to test in the next few minutes together is that some of these cells might be expressing actually quite high levels of SPC. And only a subset of these green cells are reaching all the way to the distal goal of inducing the surfactant protein C type 2-like goal that we desire. And so to test that hypothesis, next slide. To test that hypothesis, Anjali Jacob, a graduate student in my lab, and Finn Hawkins have been engineering additional colored reporters into meaningful loci in the genome. So after they induce this green sulfate decision and these cells grow out, Anjali has used Talens targeting to introduce a red fluorescence protein, the tomato reporter, into the human SPC or surfactant protein C locus, and the hypothesis I'm explaining to you is that some of the cells over time then will reach all the way to a goal where they might turn orange, co-expressing both the NKX-driven reporter GFP and the SPC-driven reporter red. And so to test that hypothesis, I'd like to, I'm very excited to show you a movie for the first time in public. I've waited a long time to show this movie. We've come a long way since 2002. The cells have come a long way too. They're now a little blurry because they're piled up. We're at 1 month in the differentiation recipe now in 3D, piled up cells that have already turned green. Lung specification has occurred. And we're waiting to see if some of the cells might turn orange. And I focus your eyes here. As you see, cells proceed through lung lineage specification, through differentiation to induce this loop of monolayered epithelial cells that are now expressing the SPC-driven reporter. And if we take a look at those cells, Anjali is able to purify just the green cells and grow these sphere-like structures that are all green. And some of those cells, in fact, are reaching this goal, but it's only a subset of the cells, perhaps 5%, that are turning red. And she's able to sort those cells out and now ask, how do their transcriptomic type 2 pneumocyte so-called markers compare to real adult lung biopsies? And if she sorts the cells out from one run, this is a sort gate shown here, you can see the transcripts of these 5 distal lung markers are now expressed at really nice high levels that in some cases are even higher than the adult lung biopsy control. So I think we're not yet comfortable from calling these cells type 2 pneumocytes. They express interesting markers. Anjali's committed to profiling the entire transcriptome of these cells, and we believe we're probably still many years off from generating a type 2 pneumocyte in culture, but I think we've also come a long way since that initial idea in 2002 of trying to do this work. So now that we've spent the time developing tools and generating some interesting cells in culture, let's try to do some work with them and apply them to our patients and to clinical questions. This is the dream we have of taking easily accessible patient-derived cells such as peripheral blood or skin fibroblasts, reprogramming them to iPS cells that we can freeze down in a repository that's now supported by an R24 as an international resource for sharing these cells. Please contact me if you want any. They're available free of charge through open source sharing. We'd like to differentiate those cells into lung lineages using some of the approaches I've shown you. And then the interesting projects begin of trying to model disease, develop drugs, or predict personalized drug responses for our patients, and maybe one day even to engineer new lungs or cells or tissues that can be delivered to our patients. So let's see if we're able to apply the whole circle now to cystic fibrosis. This is Dr. Brian Davis's important new paper just this year in Stem Cell Reports, where he's completed this circle. We're fortunate to collaborate with Brian in this project. He's used that iPS clone that I've been showing you, a line made from the child with cystic fibrosis. In this Using chamber assay, a monolayered epithelium generated from that child's iPS cells has very little short-circuit current here. And in the presence of the CFTR potentiator drug, VX809, you can see the current pick up. Oh, by the way, I'm supposed to state I have absolutely no conflicts of interest. I forgot to say that. And I didn't include the slide because I just have no conflicts. I don't know why I have to declare something that I don't have. But I have no conflicts, and this is a drug that I'm unconflicted about reporting to you. Now, the amazing data here is that using genome editing that I showed you could introduce green reporters, of course, can also introduce strands of DNA that have the correct sequence for CFTR. And so Brian has developed gene editing to essentially correct the genetic lesion responsible for CFTR. And in this iPS cell clone, the gene-corrected clone now shows this level of ion flux or short-circuit current in the laboratory dish. I think this is a very exciting figure for me, an important work, and we can imagine in the years ahead applying this model system to understand disease and predict drug responses for our patients. So, is lung regeneration an achievable mission? And how close are iPS cells to being clinically applied? I'd like to discuss with you in the last few moments. We've been doing interesting cells in the laboratory dish, but have we actually helped any patients, and how close are the cells to being in clinical trials? Well, I'd remind you they're already in clinical trials for macular degeneration. There's 2 trials ongoing that are exciting. One involves retinal pigmented epithelial cells that were derived from embryonic stem cells off the shelf. So this is an allogeneic transplant of human embryonic stem cell derived retinal pigment epithelial cells. And this is the retina of the patient that received the injection of these allogeneic cells for macular degeneration. And in the phase 1-2 data reported now, some of these patients, their vision is reported to be improved. And you might wonder, well, what about the new cells, iPS cells that might be autologous? So this very exciting announcement came in September 2014. The ophthalmologist Dr. Masayo Takanashi at the Riken in Japan announced to the world that the first patient on planet Earth had in fact received iPS cells for macular degeneration. Similar experiment or clinical trial starting, the first patient had received retinal pigmented epithelial cells derived from her very own reprogrammed cells that were iPS cells. That's very exciting. We're waiting to hear the results of this trial, and our goal is not to hear about clinical benefit. This is very early. We're just really hoping that no harm comes, that no tumors grow, and if no harm comes, that would be a tremendous success for the field from this trial. And I think trying to overhype this work and sell people that are blind now seeing is really not going to help the community. We're just trying to understand the harm these cells might do as we develop safe therapies for perhaps our children and grandchildren. So how might this then be applied, iPS-derived lung cells, for example, type 2 pneumocytes, to our patients? Could we inject cells into the lungs and actually engraft them? That's a very high hurdle, very difficult to achieve, and I don't believe there's been a believable literature previously, despite many claims, that anyone's ever achieved robust lung epithelial engraftment of an exogenously derived cell population. But on Christmas Eve on 2014 came this Nature paper that I think is important. It's from Hal Chapman and Andy Vaughan. It represents the histology, immunofluorescence, fluorescence microscopy, and immunostaining for SPC of a lung in a mouse that was damaged by influenza injury that was severe. And Hal and Andy published a new lung progenitor population they could purify out of mouse lungs. So it's an endogenous lung progenitor. It's not engineered exogenously. It's this endogenous progenitor that injected intranasally instilled into this injured mouse. Red or green cells mixed together could be seen here in the lung, and I think this is probably the most convincing evidence of successful engraftment of a exogenously delivered cell type into the lungs, and I really congratulate Andy and Hal for this achievement, and we'll see in the years ahead whether this is reproducible in other animals and might start to incorporate human cells. So the future is bright. Keep hope alive that our cells might be delivered even into lungs based on this kind of work. And I think the red and green Christmas colors are very appropriate, don't you, for a Christmas Eve publication in Nature. Very nice. To end, I'd like to retell a story I first told in public at ATS several years ago. It's to try to understand another potential of iPS cells, that to accomplish what many refer to as precision medicine, the idea you could precisely predict therapy for one individual rather than syndromically treating a cohort of patients identically. So we tried to see whether iPS cells might be used for precision medicine when we were approached in 2009 by Gordon Keller and Rocky Cass, who told us that there was a child with long QT syndrome who suffered from intractable ventricular fibrillation events. This child had a mutation in the SCN5A gene that encodes a sodium ion channel, and so the child's cardiomyocytes were electrically defective. The child would go into VFib 100 times per month, was refractory to FDA-approved antiarrhythmics, and this child had an implanted cardioverter defibrillator that would shock him to save his life, to shock him out of VFib approximately 100 times per month. So we envisioned that if we could make iPS cells from the skin fibroblasts from that child, as well as the mother and father as controls, we could produce iPS cells that could be driven forward to cardiomyocytes in vitro, and that might model the disease and help to predict drugs that the child might respond to, since he had been refractory to therapy. So the work went well, and we made iPS cells, and we shipped those cells to Gordon Keller, who, using the same concepts I've spoken about, directed differentiation, he patterned the cells through mesoderm to a phenotype that looks like cardiomyocytes, and after several days you can come to the microscope, and in a child's reprogrammed fibroblast, you can see this event, which is beating cardiomyocytes generated from iPS cells of that individual. Now, these cardiomyocytes, importantly, have abnormal electrical activity. that can be quantified. And in Rocky Cass's lab, by patch clamp analysis, one can see the delayed late sodium ion flux in just the child's iPS-derived cardiomyocytes compared to normal control iPS-derived cardiomyocytes. And that represents to us a disease model for this particular individual. And so from there, Rocky Cass was able to go on and test antiarrhythmic drugs and predict that in the in vitro model, in this child's cells, maxillatine at a low dose combined with 96 beats per minute electrical pacing seemed to ameliorate this channelopathy in vitro. And another drug the child was on, flecainide, seemed to have no efficacy in vitro, and this information was communicated to the treating cardiologist of the child. So the drug therapy predicted to work by this model was followed in vivo in the child, and the child's long QT interval was ameliorated. And then we tracked the number of arrhythmias and shocks for this child for the following 3 months. So you recall that 3 months before this therapy change, the child had 100 VFibs, 100 shocks from the ICD per month. And in the 3 months after this therapy was instituted following the child, there were recorded 0 shocks and 0 ventricular fibrillation events. This was one of the most meaningful experiences of my life, but I think the lesson here is deeper and more sophisticated than it might first appear. I just ask you to consider with me that this child who was helped at this time in this trial actually benefited not from any of our work, but from the many scientists who came before who studied frogs, worms, and Drosophila and figured out those signaling pathways that allowed the heart cells to be produced in vitro. And that kind of balanced investment in our society, in those who are curious and do basic work with absolutely no pretense at clinical application, are the true heroes that inevitably allow the clinical applications of those that follow. And I think we stand on their shoulders and should continue that investment if we're really to help our grandchildren. So in summary, I've taken you through a 40-minute journey asking you to consider whether we could enter a new era of augmenting lung regeneration, since we know the lungs can heal and repair and proliferate already. There's many ways to engineer endogenous regeneration that I've not had time to discuss. But I have tried to tell you about exogenous cells that derived in the laboratory that might be meaningful, such as induced pluripotent stem cells that arise from the invention of reprogramming and are filling up banks across the world that are available to researchers to use. I've talked to you about our now 13-year mission to try to derive de novo lung epithelial lineages from stem cells, starting with embryonic stem cells, and after iPS cells were invented, later realizing that these same recipes could be applied to either ES cells or iPS cells to differentiate lung tissue. I told you about the concept of directed differentiation and how understanding very basic early developmental biology is key to this work succeeding. I talked to you about engineering GFP reporters, such as the beautiful NKX2.1 GFP knock-in mouse that taught us some of the secrets of lung lineage specification and what that big bang of lung development looks like in mice and in the laboratory dish. I told you how those discoveries could be applied then to pattern cells that are human, that are made from iPS cells derived from our patient with cystic fibrosis and many others. And finally, I told you about the invention of genome editing and how that is transforming research and Dr. Brian Davis's successful application of editing to fix the CFTR locus in iPS cells with functional recovery in this figure. And lastly, I told you the one story of precision medicine where iPS cells at least were applied to one patient in New York City. And so I've learned a few lessons along the way that, if you'll permit me 30 seconds more, I'd like to share with you. First, I've learned that all research is translational, and if you are moved or believe my message today, I'm asking now for your help. Please, doctors, clinicians, researchers, if you see patients, it is your responsibility and duty to tell your patients, to tell the community, and to tell our leaders in Washington especially, that all research is translational and balanced investment in society is the most responsible way to help our grandchildren. I'd ask you to recall that mouse, Xenopus drosophila, and worms, C. elegans, paved the way for this work, and I'd ask you to also consider that we are in this together, and that open source sharing of knowledge and these cells and reagents is the best way forward. I'd also like to mention that I've learned that leadership matters a lot. We are fortunate to have unsung heroes that lead this community. of stem cell researchers and lung regeneration researchers. It's a wonderful community, and Zia Borak, shown here, I'm indebted to, who organized the first mini symposium on stem cells at ATS, and she has been a champion who has been telling ATS that stem cells have something to offer lung researchers from the days before stem cells were cool. Thank you, Zia. Dan Weiss is our unsung hero who leads the community and organizes the Vermont Lung Stem Cell Meeting every other year in Burlington, Vermont. He also heads the ATS Working Group on Stem Cells, and Carol and Sarah at the NHLBI have pioneered the Lung Regeneration Repair Consortium and the Lung Map that is advancing the work. And of course, where would we be without Jeff Whitsitt, our colleague and mentor, whose discoveries in lung epithelial biology and developmental biology enable all that we do. And I'd encourage you to see those discoveries on Wednesday's ATS Discovery Talk given Wednesday morning by Jeff. This is my second family. It's the lung stem cell family, meeting at Vermont every other year. I've been honored to be part of them, and I hope in some small way I've been able to represent their discoveries, our shared work, and I hope I've communicated to you our shared dreams and hope that lung regeneration is an achievable mission. Thank you.